Hello everyone, I'm Elena Gonzalez from MDPI and I will be your host today. Welcome to the first live session of the first International Electronic Conference on Molecular Sciences, Druggable Targets of Emerging Infectious Diseases. This conference is hosted online from today until the 14th of September, and it is organized by MDPI and the International Journal of Molecular Sciences. During this time, all conference papers and presentations submitted to the conference will be available for discussion on the conference website. I would like to thank our conference chairs, Professor Claudiu Supuran, Dr. Clemente Capasso, Professor Paola Gratteri, and Professor Silvia Selleri, and the conference committee for making this conference possible, and our speakers and attendees for joining us today. I would like to remind everyone that this webinar is being recorded and that the recording will be posted soon in the conference webpage. However, please note that you will have to be logged into SciForum to watch or to view the recording. Today's session is also being live streamed in YouTube. So hello to those who are watching us from there. So we will have two speakers today, Professor Jean-Yves Winum from University of Montpellier in France, and Professor Marc Ilies from Moulder Center for Drug Discovery Research, Temple University in the States. Both presentations will be followed by a Q&A session moderated by the chairs. So if you have any questions to our speakers, please feel free to post them on the Q&A below at the bottom of your screen. Now I hand over the floor to our chairs and enjoy, everyone.
So good afternoon to everyone, and welcome to this e-conference. Emerging and re-emerging infection disease in human and veterinary medicine is the title of this first session of the first session of this conference. And as you well know, infection disease represent the most exciting and dynamic areas in human and veterinary medicine. So today it's a great pleasure for me to introduce the first speaker of this session, Professor Jean-Yves Winum, who is an eminent scientist and one of my dearest friends. In 1998, Jean-Yves got his PhD in chemistry at the University of Montpellier, France, and he worked as a postdoctoral fellowship in USA and Switzerland. Currently, Jean-Yves is a full professor in the Department of Chemistry of the University of Montpellier and is an associate editor of the Journal of Enzyme Inhibition and Medicinal Chemistry. During his career, Jean-Yves published more than 200 manuscripts, and his research is mainly focused on the organic and medicinal chemistry of metalloenzyme inhibitors. And in fact, he synthesized many efficient inhibitors of carbonic anhydrases, which is a superfamily of enzymes involved in the reversible hydration of the carbon dioxide. So Jean-Yves today is going to talk about zinc metalloenzymes as potential targets against the bacterial pathogen Brucella. So Jean-Yves, you are welcome, and you can proceed with your talk.
Thank you for this very nice introduction, Clemente. Hello, everyone. I would like first to thank the organizing committee for the invitation to give this talk. It's a great pleasure for me to attend this conference online dedicated for this first session to druggable targets of emerging and re-emerging infectious disease in human and veterinary medicine. So in this context, I would like to give you an up-to-date overview of the role played by crucial zinc metalloenzyme in Brucella species and describe the approach explored to identify inhibitor with potential application as novel antimicrobial drugs. Oh, connection. Okay. So first, what are Brucella bacteria? So Brucella species are gram-negative facultative intracellular cocobacilli, which can multiply in a host cell, so I mean the macrophage, and which is capable of long-term persistence. So they are the etiological agent of brucellosis, the most important anthropozoonotic disease worldwide. So brucellosis is a zoonosis. So it means that it infects animals, and the disease can be transmitted from animals to human, but there is no transmission from between humans, or it's extremely rare. So meaning that you cannot have a pandemic like the one that you have now with COVID. So incidence of human brucellosis is reported at around 500,000 incident case each year. So the disease, it's a huge economical problem in agriculture, for livestock industries, and for trading animal between countries. So brucellosis is endemic in the countries of the Mediterranean area, Latin America, and Middle East. It has been recognized as a re-emerging disease in the developing country worldwide, as the surveillance of animal population is lacking in many developed countries and developing countries. So for example, in China, Mexico, Greece, and Brazil, the incidence of brucellosis is increasing each year. So the disease is highly debilitating, and disabling, and is characterized by flu symptom and undulant fever. So in the lack of diagnosis and appropriate antibiotic treatment, the disease may become chronic in about 50% of the case, and can cause abscesses of target organ, neurological disorder, inflammation of the joint and bone destruction, and can lead to death. Currently, antibiotics therapy consists of, in general, in a beat therapy of tetracycline and rifampicin for at least 6 weeks, and much longer during the chronic stage of infection. So a safe and effective vaccine in human is not currently available, and antibiotic strains are easily conceivable and have been isolated frequently. So finally, Brucella is considered to be a bioterrorism organism. It's classified by CDC as Class B bioterrorism agent. And this is due, in fact, to its low infection doses. In fact, with 10 bacteria, you can be infected. The capability of persistence in the environment, rapid transmission via different routes, including aerosol. And finally, due to its difficulty of treatment by antibiotic, it places significant burden on human healthcare system and limits the economical potential of individuals. So just to mention that to manipulate Brucella, you need a biosafety lab of P3 biosafety lab. So what about the pathogenic Brucella? So the genus Brucella currently consists of 12 species of Brucella, all of which are infecting mammals, 4 are pathogenic for human. So Brucella melitensis, coming from the goat, Brucella abortus coming from the cattle, Brucella suis from the pig, and in very few cases, Brucella canis. In the past decade, new species and atypical strain of Brucella have been described. For example, so Brucella inopinata, which have been described recently in human and which have no known animal reservoir. You have Brucella, so these strains in fact are fast growing, metabolically very active, and are more resistant than the most pathogenic classical Brucella species. So suggesting possible, in fact, advantage in survival in uncommon environment, food, and host. So the pathogenic potential for human and some of these species is yet unknown. For example, with Brucella microti, so in addition to its capacity of rapid replication in human and murine macrophage-like cells, it is the first species described to be lethal in murine infection. So the identification of novel, more environment-associated strains and species of Brucella raise the question of the possible risk of infection for humans and re-emergence, in fact, of brucellosis in countries considered at present as brucellosis-free. So as mentioned before, Brucella species are facultative intracellular pathogen which multiply in macrophage between 12 and 24 hours post-infection. So Brucella have developed strategy counteracting the host cell defense, resulting in fact in intramacrophagic survival. The gene required for intramacrophagic survival replication of Brucella are a subset of virulence genes of the pathogen and were called the intramacrophagic virulome. So the virulence factor of intracellular bacteria may be in fact useful target in the development of antibacterial that specifically suppress the intracellular replication of the pathogen. This approach, in fact, was validated by the group of Dr. Stefan Köhler in Montpellier, is bacteriologist, it's a close collaborator, who in fact describes the analysis of the intramacrophagic virulome of Brucella suis. So several biosynthesis pathways are absent from mammals. If you look on the slide, especially amino acid biosynthesis, and in fact the enzyme involved in this pathway can be considered as drug target. So on the basis of the complete genome sequence of the 3 human pathogens, Brucella melitensis, Brucella suis, and Brucella abortus, and on the advancement, in fact, of genomic and proteomic, so the design of mechanism-based drugs by targeting defined proteins required for the growth and virulence of the pathogen has become possible. In fact, Brucella genome encodes several metalloproteins, which start to emerge as new potential antibacterial targets and are currently under investigation, in part by our group. So you have approximately between 3 and 5% of bacterial genome which code for metalloprotein, of which several are believed to be essential for bacterial growth and survival. So this enzyme has been broadly classified into different categories depending on the biochemical pathway in which they are involved, virulence factors, resistant determinants, or essential gene. Among them, histidinol dehydrogenase has been shown to be essential for intracellular bacterial growth. I will talk about that in the next slide. Carbonic anhydrase, which are also present in many other pathogenic intracellular bacteria, such as Mycobacterium tuberculosis and Francisella tularensis, start also to be investigated in detail by our group and others in order to identify anti-infective agents with novel mechanisms of action. By the way, do not miss the next conference of Professor Marc Ilies on the carbonic anhydrase from Mycobacterium tuberculosis. So the virulome of Brucella have been defined as a whole set of genes required for its virulence. It means involved in the invasion of the host by the pathogen and in the adaptation of the environment provided by the host. So among them, the gene ISD encoding the enzyme histidinol dehydrogenase was identified essential for intramacrophagic replication. So histidinol dehydrogenase, it's an homodimeric metalloenzyme containing 1 zinc ion in each subunit. It catalyzes the last 2 steps in the biosynthesis of L-histidine, which is in fact a sequential NAD-dependent oxidation of L-histidinol to L-histidine via L-histidinol. This enzyme is present only in bacteria and in plant. To date, histidinol dehydrogenase has been cloned and characterized only in 3 bacteria, Salmonella typhimurium, Escherichia coli, and Mycobacterium tuberculosis. So the histidine biosynthesis pathway is being absent from mammalian cell. So this metalloenzyme represents, in fact, a selective and promising target for the development of antibacterial agent, avoiding secondary effect of potential inhibitor on the host. So therefore, the potential attractiveness of HDH as a target, encourages the development of HDH inhibitors. So what are the advantages of impairing intracellular implication? First, the absence of impact on the extracellular natural commensal flora of the host, limitation of the possible selective pressure on the intracellular niche, possible lower mutation rate in bacteria exposed to the inhibitor, as selective pressure, in fact, occurs only in the host cell. Novel inhibitor may be used as antibacterial adjuvant in combination with classical antibiotic, thereby diminishing, in fact, the risk of appearance of resistance to antibiotic. So the HDH encoding gene from Brucella suis was cloned and overexpressed, and protein was purified. So all this work was done in Montpellier, collaboration with Dr. Keller. The HDH encoding gene was cloned into a Escherichia coli expression vector, enabling inducible expression of the recombinant protein. The protein was overproduced as N-terminal hexahistidine tag. And then the recombinant HDH was purified by cobalt affinity chromatography. So analysis by SDH page shows that the purified protein has a mass of around 49 kilodalton, which is in agreement with the calculated value. So the yield of this purification was around 43 milligrams per liter of bacterial culture, and specific activity of Brucella suis was calculating with a KCAT about 12 second -1, and the enzyme affinity with a Km approximately of around 12 micromolar. So we developed the first generation of Brucella suis HDH inhibitors and investigated, in fact, the inhibitory effect of the series of substituted benzylic ketone derived from histidine on the enzyme activity. The data show that for some, we got effective inhibitors targeting these bacteria with inhibitory activity in the nanomolar range. The biological effect of this drug on the in vitro growth of Brucella suis in minimal medium, as well on the multiplication of the pathogen in human macrophage-like cells, was investigated. So we reported, then we reported the crystallography structure of mutated form of Brucella suis HDH, both in its unbound form and in complex with nanomolar inhibitor, thus providing the first structural background for the rational design of potent Brucella suis HDH inhibitor. So this work was realized by Dr. De Simone in University of Naples. So Brucella suis HDH, so C3C66S, contain one amino acid substitution, in particular, so the cysteine residue in position 366, which was mutated to serine. This mutant form was used for all structural studies since it avoids potential crystallization problem caused by the mixture of structural different reduced and oxidized form of the enzyme. So to get insight into the molecular basis responsible for this strong enzyme inhibitor association, the interaction of this compound with Brucella suis HDH has been investigated by means of X-ray crystallography. Analysis of the electron density map at various stage of crystallographic refinement shows the binding of one zinc ion and one inhibitor molecule in the active site. So the inhibitor can coordinate the zinc ion through its backbone nitrogen with an octohedral geometry, which was completed by the protein residue belonging to both monomer. So inhibitor binding was also stabilized by several other polar and hydrophobic interaction with enzyme active site residue. The benzyl oxyphenyl tail of the inhibitor was located in the groove responsible for NAD recognition. So encouraged by the promising HDH inhibitory activity, we start a systematic structure modification program to increase the potency and to understand the structural requirement for the key side chain by optimizing the interaction with the enzyme active site. To this end, so 3 structural type of side chain comprising parasubstitution pattern in phenylene were synthesized. So the introduction of all these different chains was based on the use of palladium catalyzed reactions such as Suzuki and Sonogashira reaction. And this preparation was achieved by 2-step synthesis, so starting from the bromo derivative, So we show that introduction of substituted aryl or substituted alkynyl chain, aryl tail, sorry, in the lead compound gives some potent and selective HDH inhibitor. Among these compounds, we got a compound with a strong inhibitory effect on HDH with IC50 of 3 nanomolar. This compound was able to impair, in fact, the growth of Brucella suis in vitro in minimal medium and also in ex vivo in macrophage host cells. In vivo tests on infected murine model show reduction of half of the number of bacteria in the spleen of the mouse 16 days post-infection with these inhibitors. So we were able to also to get to make modeling on this compound. As a result, each molecule fit well in the catalytic pocket, adopting the similar binding orientation to histidine, the co-crystallized ligand, and product of HDH. As expected, the zinc cation is coordinated with 4 nitrogen atoms belonging to the 3 imidazole moiety, H262, H419, and the imidazole moiety of the inhibitors. Interestingly, the aromatic side chain is oriented directly towards the NAD pocket, predicting a competition between the type of inhibitor and the cofactor. This region is hydrophobic and open to the solvent, predicting, in fact, a good affinity for the lipophilic part of the molecule. The aromatic side chain of both nanomolar inhibitor are oriented in the same direction of the NAD plus pocket. And on the slide, you can see the superimposition of the inhibitor with the NAD structure, which allow demonstration that an identical volume was occupied by the active site. So the molecular modeling study demonstrated the high capacity of both compound to complex, in fact, the cation zinc 2+ in the hydrophilic histidine pocket, and to reach the second lipophilic pocket where the NAD cofactor bind. So let's switch on the carbonic anhydrase. So among zinc metalloprotein encoded in the Brucella genome, 2 carbonic anhydrase encoding gene, so BR1829 and BRA0788 were identified and characterized. These 2 zinc metalloenzymes are of 25 kilodalton were shown to belong to the beta class carbonic anhydrase family and have the characteristics similar to those of other bacterial beta-CA found up to now. All conserved amino acid residue typical of beta-CA and involved in the catalytic cycle. I mean, the 4 zinc binding residue, cysteine 42, aspartic acid 44, histidine 98, and cysteine 101 are preserved. So the kinetic parameters for CA2 hydration reaction catalyzed by some human CA isozymes. So we have on the table and beta CA enzyme from Brucella suis. So Brucella suis 1, Brucella suis 2, and also I put on this table so the one for Mycobacterium tuberculosis, or the 3 carbonic anhydrase from Mycobacterium tuberculosis, and you have also the inhibition data with acetazolamide for each of these enzymes. So it was demonstrated that this bacterial metalloenzyme efficiently catalyze the CO2 hydration to bicarbonate and proton. And Brucella Swiss carbonic anhydrase 2 is slightly more active than Brucella Swiss carbonic anhydrase 1. So the first inhibition studies on these two carbonic anhydrase were reported by our group and were performed on a library of the main classes of sulfonamide inhibiting human carbonic anhydrase, including so clinically used inhibitors. So all compounds, in fact, efficiently inhibited these two carbonic anhydrase with inhibition constant between 84 around 80 and 900 nanomolar. So first inhibition of Brucella Swiss growth in minimal media with this carbonic anhydrase inhibitor was realized in vitro, and we're able to show that some of these compounds were able to reduce the growth of Brucella with a concentration of 100 micromolar. So the study was extended, in fact, to glycoinhibitors, more particularly with glycosyl sulfanilamide. And we observe a very nice inhibition constant with this type of compound, with inhibition constant around 10 nanomolar. For the first time, so a significant inhibition of Brucella Swiss growth was observed in vitro with the rhamnopiranosyl derivatives at the concentration of 100 micromolar, as well as 10 micromolar in comparison with acetazolamide. So then different carbohydrate-based Brucella Swiss were published by our group. So in 2015, you have the group of Pedro Colinas from Argentina, which published two interesting studies describing the inhibitory capacity of small library of cis-cinnamoyl glycoside incorporating a methoxyaryl or phenol moiety. So the inhibitory activity of these best carbohydrate-based carbonic anhydrase inhibitors against human CA2 and the purified Brucella Swiss CI1 and CA2 of the pathogen, so are listed in the table. All compounds show activity in the micromolar range. The most interesting feature, so they are less active than the previous compound, indeed, but the most interesting feature of these studies was the selectivity of the two most active compounds with preferential inhibition for the Brucella Swiss carbonic anhydrase over the human CA2. So the same year, our group reported glycoinhibitor in hydroxysulfonamide series obtained by Ferrier glycosylation of glycal. In this study, the glycoinhibitor were more active than on Brucella Swiss carbonic anhydrase 2 than on the isoform Brucella Swiss CA1. And among the small library of compound tested, in fact, we show that we have an interesting selectivity profile with a preferential inhibition of the bacterial over the human carbonic anhydrase isoform and inhibition of one of the bacterial isoform over the other. So in fact, these two studies show the importance and the opportunity to use carbohydrate scaffold in the design of carbonic anhydrase inhibitors. So what is the potential role of carbonic anhydrase in Brucella Swiss growth? In fact, in the first attempt to study the physiological function of both Brucella Swiss CA1 and the Brucella Swiss CA2, and their possible participation in virulence mechanism of Brucella, so the group of Stefan Keller performed separate inactivation of both Brucella Swiss carbonic anhydrase encoding genes in Brucella Swiss. In parallel, it was tried to construct a double mutant by concomitant knockout of both genes, but all this approach has been unsuccessful. So this indicates, so in fact, in the first case, when you make the mutant of one Brucella Swiss, one carbonic anhydrase, the other compensates the first one, in fact. But when you try to have a knockout of both genes, in fact, it's impossible to get the bacteria, the mutant. So this indicates that the essential character, in fact, of with at least one vital function of carbonic anhydrase in Brucella, necessitating one functional copy. Sorry. So other important work, recent important work from the group of Claudiu Supuran shows that Brucella Swiss carbonic anhydrase are susceptible to activation with amino acid and amine, with activation constant in the range of micromolar, in fact, between around 1 and 43 micromolar. So this study must be put in perspective with the recent work of Garcia Lobo group, who compare, in fact, the difference in gene content among different CO2 dependent and CO2 independent Brucella strains. And they confirm, in fact, that the enzyme responsible for this phenotype was Brucella Swiss carbonic anhydrase 2. So in fact, they demonstrate that the existence of genetic polymorphism between the both Brucella Swiss carbonic anhydrase. So to finish my talk, so I would like to say that carbonic anhydrase represent at this moment a very promising target for obtaining anti-Brucella agent devoid of resistance problem of the clinically used antibiotic. But further studies are needed, obviously, to validate this enzyme as novel drug target, because we don't know very well how its role in the growth and the development of Brucella. As non-human enzyme involved in the biosynthesis pathway of histidine, so HDH can be considered as another very promising target against Brucella Swiss. This enzyme has proven to be a virulence factor, and its inhibition constitutes an innovative antibacterial approach. So potent inhibitor, as you saw, that have been synthesized and tested, but complementary studies on their drug ability must be undertaken to evaluate the potency of HDH inhibitor as a novel drug against brucellosis. So obviously work are in progress on both of these type of enzyme, carbonic anhydrase and HDH, to identify selective inhibitors. So my group and other group. So to finish, I would like to acknowledge some people involved in this project. So Stefan Keller, bacteriologist, and Safia, former students who now are Dr. Pascal, Marie-Rose Abdo, and François Turtot, Professor Supuran, Dr. De Simone from University of Naples, Pedro Corinas from Argentina, Professor Mazurel from University of Namur, and obviously the financer of this of all this project. So French Ministry of Research, University of Montpellier, CNRS, and the Ministry of Defense, the German Ministry of Defense. Thank you for your kind attention.
Okay, thank you, Jean-Yves, for this very interesting talk and nice talk. Thank you very much, Jean-Yves. So discussion now is open, and any question about that? I don't know.
I think Claudiu has a question. Claudiu?
So, Jean-Yves, thank you very much for this great presentation. I'm used to your presentation, so it's not a surprise. My question is the following. Did you consider to use dual inhibitors of histidinol dehydrogenase and carbonic anhydrase, so the same molecule trying to target two different enzymes?
Yeah, No, for the moment, we did not work on this type of dual inhibitors. But this is a good idea. In fact, this is what we developed for other type of projects. So, you know, no, it's a good idea. We should try this,
Okay,
this type
Marc.
of strategy.
Marc, do you have a question?
Sure. So, Jean-Yves, excellent presentation. Thank you very much. My question relates a little bit to the effect of these inhibitors on the pathogen. So, can they kill the pathogen, or it's just bacteriostatic? And when can they, you know, perform the killing, and when it's, let's say, just reducing its spread?
In fact, when Brucella is inside the macrophage, Brucella needs to multiply. It needs to synthesize by itself the amino acid. So it's why they activate different biosynthesis route to have this amino acid. And so when you, in fact, when you block biosynthesis, this biosynthesis route, in the case of histidine, in fact, bacteria die.
Hi, Jean-Yves.
So you, in fact, you stop the development of the bacteria inside the macrophage.
Jean-Yves, there is a question from Fabrizio.
Okay.
He wants to know if there are any possibility of your compounds get metabolized from the host or from the microorganism themselves.
Is there any possibility your compound get metabolized? What does it mean? What does it mean? It means that compound can be inhibitor can be metabolized, and the metabolized compound is toxic for the bacteria.
Yes, I think
this
so.
This is a question.
Yes, yes.
In fact, you can use some aspect. In fact, when for the case of Brucella, so the condition inside the macrophage are acidic conditions. So maybe we could use, for example, some type of prodrug which can be released specifically inside the macrophage because of the condition inside the macrophage. This is a question. This is a question that he wanted to. So you can use the condition inside the macrophage to liberate the drug. I mean, this is possible because the conditions are different inside and outside the macrophage.
Okay. I don't know if there are other questions. No more questions?
Fabrizio answer. Yeah, it's okay.
Yeah, for Fabrizio is okay.
Yeah.
Yes, so I think we can go ahead with the talk. What do you think, Claudio?
Yeah, sure. Yeah, if Elena, it's okay, we can continue with the presentation of Marc.
Yes, of course.
Thank you, Jean-Yves. It was a great presentation, as usual.
Thank you very much.
Thank you.
Okay, so I'm very much delighted to present our next speaker, who is Professor Marc Ilies from Temple University, USA. I know Marc for almost 30 years. He was an undergraduate student at University of Bucharest, where I used to be a professor in the early 90s. He decided to work with me already as an undergraduate. Then he performed an excellent PhD study with Professor Balaban, but basically on carbonic anhydrase. In around 2000, I think he moved to USA, initially as a postdoc at Texas A&M University. Then he was a postdoc at University of Philadelphia. Then he got a position at Temple University, and he's full professor nowadays. Marc has a lot of very interesting research in different fields of pharmaceutical chemistry. I will not mention carbonic anhydrous because he will speak about that, but he had relevant contributions in gene delivery, in pyridinium derivatives, and many other fields, including anti-cancer compounds and so on and so forth. So I'm very, very pleased to introduce Marc for this nice conference that I'm sure that he will contribute very, very important topics. Marc, all to you.
Thank you, Claudiu, for this very nice presentation. It is my pleasure to collaborate with you for that long, and I hope this will continue. As Claudiu mentioned, I'm a professor in the Department of Pharmaceutical Sciences of Temple University School of Pharmacy, and also member of the Moulder Center for Drug Discovery Research. First, I would like to thank the organizers, Professor Supuran, Professor Capasso, Professor Gratteri, Professor Selleri, for giving me the opportunity to speak today. And the topic of today's lecture will be on structure, function, and inhibition of Mycobacterium tuberculosis. So, Mycobacterium tuberculosis, it's the main pathological agent that generates tuberculosis, which is a very ancient disease that basically affected mankind for over 70,000 years. So even today, it remains one of the leading causes of mortality in the world, and continues to have a huge toll on human beings. Tuberculosis, it's caused by Mycobacterium tuberculosis, which is a very interesting pathogen. Essentially, it resisted, and evolved to remain highly infectious, despite innovations in diagnostics and, healthcare issues, healthcare progress, etc. The global burden of tuberculosis remains substantial today, with over 10 million new cases and 1.6 million deaths reported yearly. So about 500,000 cases are multi-drug resistant tuberculosis, and out of which about 8.5% are expected to be extensively resistant tuberculosis. So, which is almost impossible to treat. So with nowadays armamentarium, of course. So there is an urgent need to actually design and develop new drugs to treat tuberculosis and to fight the Mycobacterium tuberculosis. Now, in terms of pathogens, the human infection, as opposed to other diseases such as plague or related infectious diseases, for tuberculosis, the human infection, it's actually essential for transmission and for the evolutionary survival of Mycobacterium tuberculosis. And what's very interesting is that Mycobacterium tuberculosis seems to lack the classical armamentarium of virulence, capsules to avoid phagocytosis, or pili, or adhesins, flagellas that other bacteria have. Also, they have various enzymes or toxins. However, despite lacking this classical virulence factors, Mycobacterium tuberculosis accompanied man since Neolithic. So why is that? Well, because Mycobacterium tuberculosis, it's first a rather hydrophobic pathogen in terms of like external capsid, and also its dimensions are actually perfectly adapted to infect the human host. So, for example, importantly, Mycobacterium tuberculosis can actually fly in very tiny droplets and essentially bypass all the innate immune system in upper respiratory tract, and reach the lower lung, where one droplet can contain 1 to up to 3, only bacteria. So very small, perfectly adapted to infect human hosts, and once it reaches this lower lung, yeah, essentially it will infect macrophages on the surface of the lung. Then these macrophages, so these macrophages in the lower lung will actually carry the pathogen inside, yeah, into deeper tissues, yeah, where more macrophages are infected, and what is generated is a granuloma, shown here. So granulomas are actually controlled by the immune system. So, you know, an individual can live with the pathogen inside its body for a very long time without being symptomatic. But, you know, under certain circumstances that are not fully understood, these granulomas can undergo necrosis, as shown here, and basically liberate the bacterium, which can actually be exhaled and can infect another person or other persons. So as you can see into this cartoon, the ability of the bacterium to live inside the macrophages, as Jean-Yves mentioned for Brucella Swiss, for example, it's essential. So the ability of the bacteria to adapt and survive inside the macrophage, in the phagosomes of macrophages, which are quite acidic, so have a typical pH of 5.5 up to 6.4, sometimes even lower, is essential for the propagation of the disease and for the pathogenesis of the bacterium. So very important pH-dependent pathways. So this pH-dependent pathways, which allow the bacterium to survive at acidic pH, may function as new drug targets, and hence our interest to look into these things. So I would say rather 15, 20 years ago, the genome of Mycobacterium tuberculosis was revealed to contain 3 beta-carbonic anhydrases that are crucial for the life cycle of the bacterium. These coded RV1284, RV3588, and RV3273. So the structure of these beta-carbonic anhydrases is shown here, and they all contain a zinc ion, basically coordinated by histidine, 2 cysteines, and usually an aspartate in the inactive form, and then in the active form, the 4th ligand becomes water, and we have the classical zinc-based catalysis of the CO2 hydration reaction. Now, so these beta-carbonic anhydrases were actually cloned, and they were extensively studied into these seminal publications done by the Japanese group of Isao Nishimori in close collaboration with Claudiu's group in Florence. And now it is known that I mean, all the kinetic parameters of these 3 beta-carbonic anhydrases are known in detail, and we know that they are like medium efficiency carbonic anhydrases, about 5 to 10 times slower than human carbonic anhydrase 2, which is an alpha carbonic anhydrase. Now, importantly, they are still susceptible to inhibition with classical alpha carbonic anhydrase inhibitors, such as acetazolamide. And you can see that we have potencies of these sulfonamide-based inhibitors ranging from about 10 nanomolar up to 500 nanomolar, which actually plead for repurposing some of the classical carbonic anhydrase inhibitors or alpha carbonic anhydrases to actually target these beta carbonic anhydrases that are found in Mycobacterium tuberculosis. So in terms of carbonic anhydrase, it catalyzes the reversible hydration of carbon dioxide, forming bicarbonate and protons. We know currently 8 carbonic anhydrase classes through extensive contributions of Dr. Supuran and Clemente Capasso, which actually worked extensively on these targets, on these pathogens too. In terms of alpha carbonic anhydrases that are present in humans, we have in terms of structure, we have the zinc ion coordinated by 3 histidines. In this case, the 4th ligand being the water or hydroxyl ion. Molecular weight around 30 kilodaltons, 15 isozymes known, very processive, some of the best catalysts evolved in nature. Zooming into the active site of alpha carbonic anhydrase, you can see clearly the difference in the coordination of the zinc ion with 3 histidine residues coordinate the zinc, the 4th ligand being the water molecule. We have the proton shuttle of the enzyme, histidine 64, and the hydrophobic pocket where the substrate CO2, which is non-polar, it's accommodated. The kinetics, it's well known, it's ping pong type. So in the rate limiting step, the zinc ion acidifies 10 million times the acidity of water, facilitating the deprotonation, and the hydroxyl form, it will attack the CO2, forming bicarbonate, which is then displaced by water, closing the catalytic cycle. The rate limiting step is the deprotonation, which is actually done intramolecularly through the proton shuttle of the enzyme histidine 64, which deprotonates the zinc-bound water through a proton wire, basically making the catalytically active form. The proton is then it's moved outside the active site through a histidine cluster. This was shown in the late 90s by X-ray crystallographic studies of various groups, including Claudiu's. In terms of alpha carbonic anhydrases that are present in the human host, so we have cytosolic carbonic anhydrases, 1, 2, 3, 7, and 13. We have membrane-bound isozymes 4, 9, 12, and 14. We have 2 mitochondrial isozymes 5A and 5B, and we even have a secreted carbonic anhydrase in saliva and milk, carbonic anhydrase 6. So all these alpha carbonic anhydrases in the human body perform critical physiological roles: transport of CO2 and bicarbonate, aqueous humor secretion, calcium mobilization from bone tissues, obviously pH homeostasis, the secretion of various fluids such as gastric, pancreatic juice, cerebrospinal fluid, etc. And of course, if we like an over activity of these isozymes can generate plethora of problems for which for treatment of which the inhibitors of these isozymes were developed. So we have 2 main classes of carbonic anhydrase inhibitors: the inorganic anions, and then the synthetic ones, sulfonamides, sulfonates, sulfamides, and many others actually. So very important in terms of sulfonamides, which still represent the main class of carbonic anhydrase, alpha carbonic anhydrase inhibitors. They bind to the zinc ion in an ionized form, and they generate a tetra-coordinated species, which it's actually very stable and reflects the great potency of these compounds. So what we have in terms of drugs in the clinic to actually inhibit carbonic and alpha carbonic anhydrases, we have aromatic and heterocyclic sulfonamides, cetizolamide, metazolamide, toxazolamide, dichlorphenamide, benzolamide. And we also have other compounds, onisamide and topiramate, that have carbonic anhydrase activity, carbonic anhydrase inhibition activity, but they're not selective. And because of that, more selective inhibitors were constantly pursued. They can be topical. So dorzolamide and brinzolamide were introduced in the late 90s, and of course, as you know, thousands of inhibitors are in the in different stages of development, with one SLC0111 produced by Supuran's laboratory, being actually in advanced phase 2 clinical trials for treatment of cancers. So it's important to know this alpha carbonic anhydrases, because if we're going to use the inhibitors to actually fight Mycobacterium tuberculosis, you know, the interference with this alpha carbonic anhydrases in the human body, it's extremely important. So coming back to the beta carbonic anhydrases in the Mycobacterium tuberculosis, so zooming into their active site, so we can actually see the same zinc ion coordinated by histidine residue to cysteines, and the water molecule. This is the active form, but this is actually shown for RV1284. For the next one, RV3588, shown in B and C, so you have the inactive form when actually the fourth ligand, it's an aspartate. So the active site actually it's opened by the interaction of this aspartate with an arginine 55, yeah, which opens the coordination of the zinc ion to water and other ligands, and therefore opens up or like launches the activity of this isozyme. Now the same type of coordination, it's seen actually in RV3273. Yeah, so again, the zinc coordinated by histidine to cysteines, and fourth ligand that can be water or can be various inhibitors, including sulfonamides that bind directly to the zinc ion, as shown here. So it's important to mention these coordination details, because what we want ultimately is to build up some selectivity between the beta carbonic anhydrases that we target in Mycobacterium tuberculosis and the alpha carbonic anhydrases. And of course, the coordination of the zinc ion is of critical importance. So as I mentioned, in the early 2000s, these beta carbonic anhydrases were actually cloned, and the carbonic anhydrase domain was actually propagated and studied, and it served to actually test the susceptibility of this beta carbonic anhydrases against the classical carbonic anhydrase inhibitors that I have just mentioned. And you can see that essentially they are still susceptible to these aromatic and heterocyclic clinically approved drugs. However, the potency of these drugs against the Mycobacterium tuberculosis 1 and 3, basically RV1284 and RV3273, it's about 10 to 100 times lower. So we need to actually develop, so the need to develop new carbonic anhydrase inhibitors was actually there. And the studies were actually emboldened by the fact that the toxazolamide, which is not a very potent inhibitor of these beta carbonic anhydrases, proved actually efficient. Yeah, so proved to be an efficient inhibitor against the bacterium through a more complex mechanism that involved this faux PR-pH regulon. Yeah? So it looks like some of these drugs have additional mechanism of action that makes them very interesting for targeting this this pathogen. And you can see here the growth inhibition of the bacterium upon treatment of etoxazolamide. It's quite impressive, and a reduction of the pH, the intracellular pH of the microbacterium upon treatment with etoxazolamide. It's also significant. So a large number of inhibitors were tested against these beta carbonic anhydrases, and here we have essential contributions from the laboratories of Dr. Supuran, Dr. Winum, Dr. Capasso. So we actually, I summarized the best ones, yeah, basically fishing up this table from a recent review that Dr. Supuran actually authored. So these are the most successful compounds that are able to inhibit the 3-beta carbonic anhydrases with basically nanomolar potency. And they constitute essentially lead compounds for further studies. So we have here, for example, the ureidosulfonamides that are very potent alpha carbonic anhydrases. They can also be nanomolar potent, yeah, some of them against beta carbonic anhydrases of Mycobacterium. We have mono and DTO-carbamates, yeah, again, super potent. Yeah, so representative structures shown here. We have halogenated benzolamides, structure shown here, that we initially developed for targeting alpha carbonic anhydrase, also very potent, I mean, relatively potent against beta carbonic anhydrases. And we have the cinnamoyl glycosides. Jean-Yves actually presented some representatives, and they can be also medium potent, shown here. And finally, we have this triazinyl sulfonamides recently introduced that can also target, the 3-beta carbonic anhydrases with nanomolar potency. So if we look a little bit towards the structure of this nanomolar potent inhibitors, we can actually see that we have the zinc warhead, yeah, either benzene sulfonamide or thiazole sulfonamide or novel zinc binding groups, very powerful, yeah, attached via polar groups, yeah, that can be aliphatic or aromatic, but the general leitmotif is that they are extremely polar. Yeah? So, and the linker is very short. So the structure must be compact and highly polar. So that alluded towards the repurposing of a class of sulfonamides that I synthesized with Claudiu at the beginning of our collaboration, basically pyridinium sulfonamides. So pyridinium sulfonamides we selected at that time because pyridinium has several specific advantages: a flat structure, pi electrons, can make it fluorescent, a permanent positive charge, so they are positively charged compounds, and it's a delocalized charge, redox properties, and it's biocompatible. So we have many natural products, NAD+ and ADH. We also have drugs such as omeprazole that involve pyridinium intermediates, risedronate, and we have natural compounds, as two mentioned here. So at that time, we were targeting the carbonic anhydrase 4 isozyme, starting from the observation that this positively charged sulfonamide, quaternary ammonium benzoyl sulfonamide, can actually act selectively against CA4 since it's membrane impermeable. So so the idea was to replace the ammonium group with a pyridinium one, which can be introduced via reaction of pyrrolium salts and primary amines, the Bayer-Picard reaction, detailed here in terms of mechanism, and the pyridinium group, the pyrrolium salts needed for this purpose, can be actually synthesized by the Balaban-Nates-Cupride reaction of deacylation of alkenes or monoacylation of methyl oxide. So more recently with Chiati Dave, we built up a large library of this pyrrolium salts, which allows us to explore different substitution of the pyrrolium and therefore pyridinium salts, either 2,6, building steric bulk in 2,6, in 2, 2,6 with phenyl substituents, 2-phenyl substituents, and other different groups. So that time, we targeted the alpha carbonic anhydrases with a library of pyridinium benzene sulfonamides, and these prove very potent, much more potent than the parent aminosulfonamides. So the pyridinium group can actually enhance the potency, which is very important. The QSAR was established. These compounds were translated all the way to animals, and they proved very good in inhibiting the renal carbonic anhydrous 4. Then these studies were followed by similar studies where pyridinium benzolamides were synthesized and tested. So again, very good results, nanomolar range of inhibition for these alpha carbonic anhydrases, C1, 2, 4, and then later 9 and 12, as you're going to see. So all of them are membrane impairment. So we have selectivity for membrane-bound isozymes without affecting the cytosolic ones, the cytosolic isozymes, which can be actually an advantage when we are targeting the mycobacterium. We don't want to actually generate side effects by involving other cytosolic carbonic anhydrases. Further studies from Claudiu's lab reveal the fact that these two types of pyridinium sulfonamides actually generate isozyme selectivity through specific targeting of two main pockets. Yeah? So one pocket, it's actually shown here. The pyridinium group generates basically pi-pi stacks with phenylalanine 131, stabilizing the inhibitor. So that's why it's much more potent, the parent benzene sulfonamide. And in the case of the pyridinium benzolamides, it looks like the pyridinium group, it's actually located in a totally different area. So you can see here, and you can see that in the case of pyridinium benzolamides, we are targeting this area in alpha carbonic anhydrases. So it is possible through the pyridinium substitution to actually induce isozyme selectivity, profiting from the local topology of the isozymes. So that emerged in mid-2000, and essentially many of these pyridinium compounds were reassessed on carbonic anhydrase 12 and 9, with excellent results. And essentially, this is the detail of these lead compounds. Based on this X-ray crystal structure, one can see that potentially a better, a more potent and selective pyridinium derivative can actually be done if the pyridinium group it's moved into this particular area here by attaching it on the third position of the benzene sulfonamide warhead. And that was done basically through a collaboration of Claudiu's group with my group, and Süleyman Akotsak and Özlem Güzel Akdemir worked on this project. So we synthesized 26 pyridinium derivatives of 3-amino benzene sulfonamide via an improved microwave based reaction. So the whole reaction is done in 15 minutes via microwave, and we can access these compounds in high yield. In terms of inhibitory activity, so they're again better inhibitors against alpha carbonic anhydrases than parent aminosulfonamides, and the SAR was established. In terms of isozyme susceptibility, for the alpha carbonic anhydrases, we have CA9 and 12 as the most susceptible, followed by CA2 and CA1. As you're going to see, these compounds are very potent against the Mycobacterium tuberculosis beta carbonic anhydrases too. So the actual SAR. It's I mean the actual inhibition table. It's presented here. I apologize for this complicated table, but if we want to highlight certain things, we can see that we can achieve sub-nanomolar, so very low nanomolar potency against the two key beta-carbonic anhydrases with these new pyridinium compounds, while still having potency against alpha-carbonic anhydrases, key alpha-carbonic anhydrases. If we look into other potent compounds, so we can constantly see the same thing, low nanomolar inhibition of this beta-carbonic anhydrases with compounds that contain a pyridinium substitution with small substituents, so low steric bulk, methyls, ethyls. In the case of phenyl, we can place the phenyl substituents in the fourth position, sometimes in the second position, but not, I mean, there are limitations here. So once we increase the steric bulk, you can see that the potency decreases. So for example, the N-butyl derivatives, so derivatives that have N-butyl, they are essentially 50 times less potent than the methyl analogs, and this is constant throughout the study. So as we go from methyl to N-butyl, so we have a drop in the potency against the beta-carbonic anhydrases, and so I mean that's the same thing observed for the third set of compounds. So basically, these new pyridinium sulfonamides that were just basically published last year are very potent against the beta-carbonic anhydrases. And, we started to look into molecular modeling into their binding into the alpha-carbonic anhydrases. So this is actually published. And we are currently doing the same thing for the beta-carbonic anhydrases that against which these compounds are very potent. So that's work in progress, and I will end with the conclusions of this study. So we have 26 new pyridinium sulfonamides derived from methanolide, made through this new microwave assisted chemical synthesis. They are very potent, some of them against RV1284 and RV3273, and also against many alpha-carbonic anhydrases from the human host. Now, so the pyridinium moiety, generates increased potency, so we reach low nanomolar levels of inhibition against the beta-carbonic anhydrase from Mycobacterium tuberculosis, some of the most potent inhibitors into this against this pathogen. But the isozyme selectivity was relatively low, so that remains to be further optimized. Based on the structure activity relationships drawn, we'll continue the studies through molecular modeling and through other types of studies. So I would like to thank the people that did this, that contributed to this work, especially Suleiman, now an associate professor in Turkey, Kiati Dave, which works in Merck, Utpal Mondal, who moved to Fort Detrick National Cancer Institute, and now also moved to the government, and especially to Claudiu Supuran and all his group, for the constant collaboration, like he said, lasted almost for almost 30 years. And to you for your kind attention. I will take any questions.
Thank you, Marc,
for
this excellent presentation. We enjoyed very much. Let us see whether we have questions. So, in the meantime, I will make a comment and also a question for you. Of course, what you showed on the pyridinium moiety for inhibitors of carbonic anhydrase, it's great, and we have indeed very good data. The main issue with the bacterial enzymes is that these compounds are membrane impairment, so they can act very well for the transmembrane tumor-associated carbonic anhydrases, which are transmembrane out of the cell, but for the pathogenic bacterial enzymes, it's a problem. Did you think of procedures, for example, to do prodrugs, for example, to reduce them to the hydropyridines and things like that? Because in this way, we may have a prodrug which can cross the plasma membranes and eventually get oxidized within the pathogenic cell.
So a very good point. Yeah. So how we actually reach the bacterium, that's very important. And there are many strategies. So, for example, my group, as you know, it's very active towards designing pyridinium for nucleic acid delivery, and you know, we actually know how to tune the lipophilicity to and other molecular parameters to actually cross the membrane when it's needed, and that is definitely on the table. As you mentioned, the you know, redox prodrugs. You know, following Nick Bodor's strategy, those are also on the table. We actually tried with Suleiman this approach, and we do have preliminary data. The main issue here is selectivity. So it's very hard to selectively reduce these compounds, yeah, and you get very complex mixtures that you know, are a little nightmare to separate. I mean, we can still try to, you know, improve, you know, the strategies for the selective reduction, but as I mentioned, you know, Suleiman tried about half a year without much success here. You know, we still can do things. Yes, targeting the Mycobacterium tuberculosis inside that vacuole where he actually lives, inside the macrophage, is probably the hardest thing.
Great. Let's see whether we have any other questions from Clemente, Jean-Yves, Paola, Silvia, and the other participants. Do you have some curiosity or issues to ask to Marc?
Claudio, can you hear me?
Sure, yeah, Clemente.
Yes, Marc, great talk. Thank you very much. So I have just a question, just a curiosity. So the inhibitor you presented resulted in excellent inhibitors when tested in vitro. So I mean on the enzyme. But have you an idea of the amount of inhibitors you need to block the microorganism growth? Have you this evidence about that?
Well, so this is, as I mentioned, the translation of all these inhibitors, all classes, is the big problem here. Yeah. And you saw that even for, I mean, Abramovich tested toxazolamide, right? And he saw that, for example, this relatively weak inhibitor for Mycobacterium tuberculosis beta carbonic anhydrases can actually achieve very good results. Yeah.
Yeah.
Why? Because you know, probably the drug can actually reach, yeah, in large amounts or in significant amounts the microbacterium where it lives into those phagosomes. Yeah, so you know, it's very hard to test these things because, as Jean-Yves mentioned, you need a biosafety level 3, you know, at least to play with you know, these things. So you know, I would say the main interest was just to, you know, first generate some potent inhibitors, then to try to optimize the selectivity, if possible, between the beta carbonic anhydrases of Mycobacterium tuberculosis and the alpha carbonic anhydrases in humans. And you know, as Claudiu mentioned, you know, then try different prodrugs, different delivery ways. So my lab, I have a very big interest into these translational issues and delivery. You know, in this case, the big problem is that,
yeah,
you know, if you talk about Mycobacterium tuberculosis, then, you know, they ask you to show you the BSL-3.
Okay, thank you very much, Marc.
Sure.
I can intervene. So, you know, till recently, there was quite some reluctance from many members of the scientific community regarding carbonic anhydrase inhibitors as anti-infectives. Fortunately, last year, there was some very relevant papers from another group, not from our group, because always these papers are from our group. So, but this time it was a group from USA, Daniel Flaherty, and they showed, for example, for vancomycin-resistant enterococci and some other bacteria that it's possible to inhibit them with carbonic anhydrase inhibitors in vivo. So these are quite relevant translational studies which have been published. Now we collaborate with this group for almost 1 year, and we also have quite interesting results on this area. So basically, I think that the field of the antibacterial carbonic anhydrase inhibitors, it's still in its infancy, but we have a lot of important advancements. And what Marc showed here for Mycobacterium, it's of course very, very important, but still we are still at the point in which we miss the in vivo data, which I hope sooner or later it may be generated. Clearly, as Marc mentioned, it's very difficult to have a laboratory which can test in vivo this type of thing. So I think, for example, models like the zebrafish, because these embryos are quite small and they can be grown without any problems, and they can be infected with Mycobacterium marinum, which is not Mycobacterium tuberculosis, but anyhow, it's a good animal model for this time. So anyhow, I think we have relevant data from what Marc showed, what Jean-Yves mentioned earlier for Brucella, which is anyhow a similar organisms, let's say, from the pathogenicity point of view. And clearly, with Clemente, we are in a process of trying to have models of this type of antibacterial carbonic anhydrase inhibitors in order to validate them. So, okay, I think I spoke quite a lot. Let's see whether we have some other questions from other participants to the meeting. If not, Elena, I think we can stop this afternoon session, which was quite successful. I wish to thank both speakers for their wonderful presentation, which I think they performed a great job to the scientific community and to our nice electronic conference.
Thank you, Claudiu.
Okay.
And thanks for the organizers. Big thanks for the organizers.
Well, thank you. Before we go, I would also like to thank our chairs and our speakers for interesting contributions to this webinar. All the attendees for joining us today. There's the second live session tomorrow at 3:30 p.m. CST, like us today. So we hope to see you all there. So we will see you tomorrow. It will be Anna, my colleague here. And now I will close the live session. Good night, good evening, good morning, wherever you are in the world. And hope you enjoyed the live session. Bye-bye.
Thank you. Bye-bye.